Hello friends, welcome back to a new video that is MCQs on immunology, and this is the part 2 of this series of videos on immunology. And so let's start the question answer session on this topic. So the first question of this video, that is question number 21. So it is, in agglutination reaction, the antigen is a option A whole cell, option B soluble molecules, option C viruses, or option D protein. So in agglutination reaction, the antigen is a option A, that is whole cell is act as a antigen in agglutination reaction. So what is agglutination reaction? Here we will see this agglutination is a serological reaction that causes particles to clump together. So this reaction occurs when antibodies react with antigens on the surface of particles. So the clumping is usually a response to a specific antibody. So against this antigen, which is the whole cell, the antibodies bind to them and clump together. So we get a precipitate of particles. That process is known as agglutination reaction. So let's move on to question number 22. That is, the ability of an organism to resist infection by the pathogens is called option A infection, option B hypersensitivity, option C immunity, or option D allergy. So which one is correct about the ability of an organism to resist infection by the pathogens? And the right one is option C, that is immunity. So this is called immunity. So what is immunity? The ability of an organism to resist infection by the pathogens. That is called immunity. So next question देखेंगे. Next is question number 23. Which of the following compounds is not found in tears? And options are option A lysozyme, option B lactoferrin, option C IgA antibody, or option D IgE antibody. So which one is correct about the antibody or compound which is not found in tears? And the right answer is option D, that is IgE. So except this IgE, all other compounds, that is the lysozyme, lactoferrin and IgA, we can get it in tears. Okay, so next देखेंगे. Next is question number 24. So name the first cell which is recruited at the place of infection, and the options are option A NK cells, option B basophils, option C neutrophils, or option D macrophages. So the right answer is option C, that is neutrophil. So neutrophil is the first cell which is recruited at the place of infection. Okay, next is question number 25, that is the hypervariable regions responsible for antigen binding is contributed by option A light chains only, option B heavy chains only, option C a part of light and heavy chains both, or option D light chains and disulfide hinge. So in hypervariable region of antibody which bind with the antigen is composed of which one? Option C, that is it is composed of a part of light and heavy chain. So here we can see this is the antibody structure. Here we will zoom it. So here you can see this is a antibody structure, and this blue part is the heavy chain and this yellow part is the light chain, and the hypervariable region is here. This portion is the hypervariable region of both light chain and heavy chain, and that's why it is composed of this hypervariable region is composed of light chain and heavy chain where the antigen binds with this antibody. Okay, so this here we can see. So hypervariable region are domains on immunoglobulin heavy and light chains variable region that are in direct contact with antigen and are frequently mutated to allow diverse antigenic specificities to be recognized. Okay, So next is question number 26, that is Jenner prepared the vaccine for smallpox by using option A attenuated pathogen of the smallpox, option B smaller doses of smallpox virus, option C attenuated from form of cowpox virus, and option D material from cowpox pustules. So which one is correct about the smallpox virus? So sorry, smallpox vaccine. So the right answer is option D, that is material from cowpox pustules. So Jenner prepared the vaccine for smallpox by using material from cowpox pustules. Okay. So next question देखेंगे. Next is question number 27, that is antigenic determinant sites bind to which portion of an antibody molecule? And the options are option A light chains, option B heavy chains, option C intermediate chains, or option D both A and B. So already we have discussed here. So the right answer is option D, that is both A and B. This antigenic determinant site means the epitopes or the antigens which are sorry, the portion of hypervariable region. So here this is the antibody, this is the hypervariable region where the antigenic determinant site are present, where the antigens bind. So this portion is composed of light chain and heavy chain. Okay, so both A and B is the correct one. So let's move on to question number 28. That is opsonization is carried out by option A IgM, option B IgG, option C IgA, or option D IgE. So the right one is option B, that is IgG. So opsonization is carried out by IgG antibody, that is option B. And this opsonization is an immune process which uses opsonins to tag foreign pathogens for elimination by phagocytes. Without an opsonin like antibody, the negatively charged cell wall of the pathogen and phagocyte repel each other, so they cannot bind to each other. So to bind, these opsonins help to bind this pathogen with the phagocytes to remove those pathogens from the organ or body. Okay, so this is called the opsonization process. Let's move on to question number 29, that is T cells have a life of option A 4 to 5 days, option B 4 to 5 weeks, option C 4 to 5 months, or option D 4 to 5 years. So the right answer is option D, that is 4 to 5 years. So T cells have a life of 4 to 5 years. Okay. So next question देखेंगे. Next is question number 30, that is interferons is protein that option A inactivates a virus, option B protects unattacked cells from virus, option C prevents viruses from taking over the cellular machinery, or option D all of the above. And the right one is option D, that is all of the above. So interferon is protein that inactivates a virus, protects unattacked cells from virus, and prevent viruses from taking over the cellular machinery. So all are correct about the interferons. Okay, and these interferons are proteins that the body produces to fight against viruses. So they are secreted from infected cells and activate the innate immune response. These interferons got their name because they interfere with viruses and keep them from multiplying. Okay. So next is question number 31. So study of interaction of antigen and antibody in blood is option A hematology, option B serology, option C cryobiology, or option D angiology. And the right one is option B, that is serology. So study of interaction of antigen with antibody in blood is known as serology. Okay. So serology is a scientific study of blood serum and other bodily fluids. In practical immunological terms, serology is the diagnostic identification of antibodies in the serum. Okay, so let's move on to question number 32. That is antibody formation and immunity production by globulin protein is found in option A hemoglobin, option B blood platelets, option C plasma, or option D cytoplasm of RBCs. So the right answer is option C, that is plasma. So antibody formation and immunity production by globulin proteins is found in plasma. Globulin proteins means antibodies. Okay. So next is 33, that is antigens are found option A inside cytoplasm, option B inside nucleus, option C on in nuclear envelope, or option D on cell surface. So antigens are found on option D, that is on cell surface to bind with the antigens. Okay, sorry, antibodies. So let's move on to question number 34. Antibody production is associated by option A monocytes, option B leukocytes, option C lymphocytes, or option D erythrocyte. And the right one is option C, that is lymphocytes. So antibody production is assisted by lymphocytes. Okay, so next one is question number 35. It is an antiviral substance produced in response to viral infection for restricting its multiplication is option A antigen, option B antivirion, option C interferon, or option D virion. So which one is correct? The right one is option C, that is interferon. So an antiviral substance, that is interferon, produced in response to viral infection for restricting its multiplication. Okay, so next one is question number 36, that is which of immunoglobulin present in colostrum or mother's milk, and the options are option A IgA, option B IgG, option C IgD, or option D IgE. So right answer is option A, that is IgA antibody we will get or present in colostrum or mother's milk. Okay, so next one is question number 37, that is an autoimmune disease is option A AIDS, option B hemophilia, option C asthma, or option D myasthenia gravis. And the right answer about the autoimmune disease is option D, that is myasthenia gravis. So this myasthenia gravis is a chronic autoimmune disorder in which antibodies destroy the communication between nerves and muscles, resulting in weakness of the skeletal muscles. So myasthenia gravis affect the voluntary muscles of the body, especially those that control the eyes, mouth, throat, and limbs. Okay. So next question पे जाएंगे. Next is question number 38. That is passive immunity is option A inherited, option B obtained through vaccination, option C acquired through infection, or option D obtained through sera of other animals enriched with antibodies. So the right answer is option D, that is obtained through sera of other animals enriched with antibodies. So passive immunity is obtained through sera of other animals enriched with antibodies. Okay, and this passive immunity is provided when a person is given antibodies to a disease rather than producing them through his or her own immune system. So in passive immunity, the antibodies are given from outside, and a newborn baby acquires passive immunity from its mother through the placenta. Okay, so this is all about passive immunity. So next one is question number 39. Gamma globulins are synthesized in option A lymph and lymph nodes, option B liver, option C bone marrow, or option D kidney. So the right answer is option A, that is lymph and lymph nodes. So gamma globulins are synthesized in lymph and lymph nodes. But if we say the B cells synthesized in then we will say that B cell synthesized in bone marrow, but antibodies or gamma globulins are synthesized in lymph and lymph nodes. Okay. So the last question of this video, that is, which is not immunological technique? And the options are option A, antibiotic injection, option B, tetanus toxoid, option C, gamma globulin, or option D, smallpox vaccination. So which one is not immunological technique? And the right one is option A, that is antibiotic injection is not immunological technique. Okay, so these are all 20 questions of this part 2 video on immunology. So thank you for watching this video.